Good morning, BIO78, or whenever you're watching this, good afternoon, good evening. Welcome to our dispersal and gene flow module. So in the previous module, we focused on populations as single entities, and in this module, we are going to start to investigate how different populations interact with each other through processes such as dispersal and gene flow. So when thinking about how this topic might relate to your everyday life, if you've ever driven on a highway, you might relate to this study that I have referenced here, looking at whether California highways act as barriers to gene flow for ground-dwelling mammals. In particular, this study was looking at coyotes. And we'll talk in depth in the next couple of videos about what gene flow is, but in general, it's this movement of individuals between populations, and then those individuals are reproducing and producing offspring. So gene flow really is the movement of genes flowing between populations, right? And in this particular study, which I'll put as a link on Canvas, as far as I can tell, it's a preliminary study. It's not a published paper yet. But what they were looking at is whether 4 highways in California, they looked at 2 in the Bay Area, 580 and 680. 680 is what I take when we go visit my husband's parents in San Jose. But in the Sierra foothills, they were looking specifically at I-80 and US 50 to see if those particular freeways or highways were actually preventing gene flow of coyotes for populations found on one side of 80, for instance, versus the other side. So they collected actually scat samples from coyotes from both sides of these different freeways. They actually used microsatellite markers, which we talked about before, to try and estimate genetically whether there is gene flow occurring across these highways or whether populations on both sides of those highways are separated because of the barrier. So that's one example of how gene flow might be happening right around you in your everyday life without you even realizing it. So in this particular module, in terms of what you'll be able to do after completing this module, first of all, you'll be able to compare and contrast the effects of natural selection, gene flow, and drift on natural populations. So we'll talk in this module about how those different processes interact with each other and how we know which one might take precedent over the other one. You will be able to identify how many non-a priori populations are present in a data set. And I'll explain what non-a priori means, but basically it's where we're not using any prior assumptions or prior knowledge. We're letting the data tell us how many populations there are that are present. The third learning objective is that you'll be able to differentiate patterns of isolation by distance, or IBD, and isolation by environment, abbreviated as IBE. So we'll talk about what the different effects of those processes are and what they look like in populations. And then lastly, like we've been doing in previous modules, you'll be able to choose a scientific paper that could be used to write a proposal about dispersal and gene flow. So for the main assignment for this module, in addition to the mini quizzes, just like we've been doing, I will ask you to find a paper related to dispersal and gene flow. This is one example of a recent paper I found published in August. Again, the search terms I used were gene flow and molecular ecology. And I came up with this cool paper looking at Phyllobates poison dart frogs, which are shown here. And in this paper, they were looking at how gene flow could be affecting the phenotypes of these poison dart frogs. They're actually different phenotypes. So phenotypes are the colors. This guy's bright yellow. There are different colors of this species, and they were looking at how gene flow might be affecting that. And that's something, again, we'll talk more about later in this module as well. And again, thinking about why this is important for us, remember we have our big final proposal at the end of the semester, and then we're doing 2, I'm calling them mini or practice proposals throughout the semester as well. And that proposal 1 has to be focused on some topic that we've covered in lecture so far. This topic of dispersal and gene flow really comes right on the cusp of Proposal 1 and Proposal 2. So if you wanted to focus on a topic related to gene flow for Proposal 1, you can. If you're really interested in it and you want to use it for a Proposal 2 topic, that's fine too. So there's some leeway there because this is coming right on the border of when your Proposal 1 is due. So again, we're focusing on the skills of proposal writing to prepare you for what I'm asking you to do in this course, but also for proposal writing or similar type of scientific writing you might have to do in future jobs, future careers after Sac State.